Okay, thank you, Linda, for the introduction. So good morning, everyone. Welcome to the webinar on the important elements of cleaning validation and the health-based exposure limit. Okay, when it comes to cleaning validation, so the common questions can be, how do I design robust cleaning procedures to ensure that my equipment can consistently meet the acceptance criteria for the maximum allowable product, the residues carryover. And also some of the questions can be related to the selection of the representative products for the cleaning validation, because some facilities can have a lot of products. So how do I select the representative or the worst case products for the cleaning validation? Of course, the health-based exposure limits, I'm sure this will attract a lot of interest as well. So typical questions can be, how do I get the PDE values for the products and also the cleaning detergents, if you are using the cleaning detergents, and how to apply these PDE values in the calculation for the acceptance criteria for cleaning validation. So basically the goal for this webinar is to understand some of the important elements of the cleaning validation and also the health-based exposure limits. So current situation, so many manufacturers produce products in multi-product facilities where there is a risk of contamination and cross-contamination. So this is especially so for those generic drug manufacturers or the contract manufacturers, where there can be many products made using the same equipment in the same facilities. So under those situations, the risk of contamination is quite high if they do not have a robust cleaning procedures. So identifying cleaning validation risks and implement the appropriate controls will be very important. So such as setting the HBEL limits for the various products to mitigate the contamination risks. So in today's agenda, basically we will summarize the main elements of cleaning validation. And then my colleague Paul will continue to talk about the health-based exposure limits. I'm sure this section will attract a lot of interest. So he will talk about the regulatory timeline for the HBELs, and also what are the documentation required to define the HBEL, and how do we apply these health-based exposure limits in the calculation for the acceptance criteria for cleaning validation. Of course, the benefits to the participants will be to revisit the critical elements of cleaning validation. It's kind of a refresher, and will be able to understand the history of HBELs in cleaning validation, and also assess whether you have enough supporting documentations to determine the HBELs and to apply the HBELs in setting the cleaning validation limits. So first, we go through this section on the important elements of cleaning validation. So this is just a brief introduction to some of the regulations and the guidance documents related to cleaning validation. So back to the year 1993, I'm sure everyone will know this is the US FDA versus the Bar Laboratories, whereby there is a lawsuit against the Bar Laboratories for the serious violations of GMP manufacturing practices. So this includes the cleaning validation as well. So as a result, this leads to the FDA guidance for the inspection of the cleaning processes to ensure that the companies follow strictly the cleaning requirements to minimize the risk of product cross-contamination. Of course, in the year 2004, FDA also come up with the guidance on the risk-based approach and the PAT. So PAT is basically the process integrated technology, is to helps to measure and monitor the critical process parameters to ensure that the process is within the control. So this applies to cleaning validation as well. Of course, ICH, there's a quality risk management, and also ISP has a risk-based manufacturing documents as well. So all these risk-based principles are also applicable to cleaning validation. So next, we look at the PDA technical reports. So I'm sure all of you should be very familiar with the technical report number 29 and number 49. So this is basically related to the cleaning validation. So very soon, these two technical reports will be combined into one technical report, which is technical report number 29. So in this new revised technical report, basically this will apply to various manufacturing processes related to the cleaning validation. So this will basically come out very soon in the near future. In 2011, FDA also have the process validation guidelines. So the principles in the process validation guidelines, their various stages, is also applicable to cleaning validation. So I will elaborate a little bit more on this during the presentation. Of course, a few years later in the EMA guidance on the using the HBEL in shared facilities. So since then, the requirement for using the health-based exposure limits is implemented for the pharmaceutical manufacturing processes. And recently, of course, you also have the ASTM guidance documents on the cleaning process development and validation, and also specifically on the derivation of the health-based exposure limits. In ISP guide as well, there's cleaning validation lifecycle. So all these guidance documents are valuable resources that you can refer to if you want to know more about the cleaning validation and also the health-based exposure limits. So first, we just look at the cleaning validation, based on the guidance document. Basically, cleaning validation is a documented evidence that the cleaning procedures will consistently reduce the active pharmaceutical ingredient. So consistently means you need to prove it with 3 consecutive runs of cleaning validation to reduce the API, process residues, cleaning agents, if you're using the cleaning agents in your cleaning process, and also the microbial residues from the product equipment surfaces to acceptable levels. Because cleaning, it's impossible to remove everything completely. As long as you can remove it to the acceptable levels for the processing of the next drug products, it should be acceptable. So earlier I mentioned about the process validation guidance. So the principles mentioned in the process validation guidance is also applicable to cleaning validation. So in the life cycle approach to cleaning validation, there are basically 3 main stages. So in stage 1, we look at the process design. So this is basically to gain the knowledge. So in this stage, we look at the cleaning methods that we have. So what are the process equipment that we're going to clean? Residue detection methods, what methods we're going to detect the process residues after the cleaning. And also you need to decide and work out the acceptance criteria on what is acceptable limits for the product carryover. So all this will need to be basically incorporated here. And very important also, you need to work out the critical cleaning parameters. So, so for that, you will need to understand the type of soil that you have, and also what are the cleaning parameters that can effectively remove your product residues from the equipment surfaces. So this is a very important stage. It lays the very good foundation before you go into your cleaning validation, which is in stage 2. Of course, the stage 3 is basically the post-validation stage. So in this stage, the main aim is to ensure that your validated cleaning procedures continue to be in the validated state in the foreseeable future by having a periodic review of your cleaning procedures, and also implement the process controls like for example, the change control system deviations, CAPAs, to ensure that you continue to monitor the performance of the cleaning process, and also some of the continuous monitoring processes as well. So this is a very important stage. So in the life cycle approach to cleaning validation, the focus is more on the stage 1 process design. Make sure that you design very robust cleaning procedures. And also in stage 3, make sure that you continue to monitor the performance of the cleaning process. So basically, this is all risk-based, focus on the product impact and the patient safety. So for the cleaning process design, it's very important that you need to know the process flow of your manufacturing process, whether your manufacturing process is how many equipment train are there, and also the process equipment, whether is it a standalone equipment or the equipment is in a process train, and also the limitations in the cleaning process, whether there's a CIP process or manual cleaning process. So all this will need to be taken into consideration when you perform the cleaning process design. Of course, very important is the process soil. So there are many different kinds of process soil in the manufacturing facilities. Some are very easy to clean and some more difficult to clean. So you will need to understand the difficulty of cleaning for your various process soil. And the components of cleaning. So if you are using the detergent, so for the cleaning process for the most difficult to clean soil, you need to understand what are the parameters that can affect the cleaning process. So this basically lays the foundation for the design of a robust cleaning process. So very important is also to identify the sources of contamination. So in any cleaning validation, you need to understand what are the residues that you are going to clean. So these are the potential residues for consideration. Of course, very important is the active pharmaceutical ingredient, the drug substance. So this is the most important part. Of course, there are other excipients as well, which is equally or sometimes even harder to clean than the API. For example, some of the excipients in the carpeting, dyes, colorants, fragrance, for example, the titanium dioxide or enteric coatings. So those excipients can stick readily to the surface and not easy to remove. And also you need to take into consideration the degradants, impurities, even the lubricants, antifoams, some process, for example, the fermentation process, some antifoams which can be difficult to clean. They can form a air-liquid interface line at your vessel. And also you need to consider the bioburden, endotoxin, and mycoplasma prions. So all this can be considered as the potential residues for the cleaning process. And the worst case soil is basically based on the cleanability, how easy to clean the soil with a set of cleaning parameters. And also need to consider the toxicity of the soil as well. So highly hazardous products. So you need to look at the PDE values as well and other toxicity values to determine which is the most hazardous products. Solubility in water or in the cleaning agent as well. So this can give a criteria for the selection of the worst case soil. And the condition of soil as well play an important role. So the amount of soil on the surfaces, of course, you have more soil on the surface. So for example, if you have a campaign run of 10 pages, of course, sometimes you make the 10 pages. After 10 pages, the amount of soil on the surfaces can be more difficult to clean than compare you just make a one batch. The nature of the soil on the surfaces can be freshly deposited, which can be easier to clean. Can be dry during the process, or you have a dirty hole time. The longer the dirty hole time, the more difficult to clean. Baked on the surface as well. Compacted if you have a carpeting punch. So those are compacted soil. So this can create some cleaning issues. So of course, the cleaning process design lab scale study is very important to help you to design and optimize the cleaning procedures and a set of parameters for achieving the consistent cleaning results. So typically the lab scale studies involve coating the soil on the coupons and then simulation of the condition of the soil in the lab by using the oven. So for example, if the soil is exposed to 24 hours, 80 degrees in the manufacturing process, then you can put the soil coupon into the ovens for 24 hours at 80 degrees. And then after that, you simulate the cleaning process by using a set of cleaning parameters using various concentration of the cleaning agent, cleaning time, temperature as well. And then after that, you assess the acceptance criteria on the cleaning effectiveness for each set of the cleaning parameters. So the criteria can be visible residues. Water break-free test means you put a layer of water on the surface, make sure there's no water break on the surface. If there's a break in the water layer, then there may be residues on the surface. And also you can consider checking the weight of the coupons before the coating and also the weight of the coupon after the cleaning. So make sure they're similar in weight. So of course the cleaning parameters can be at the time very important, time of the cleaning. Action as well. Action refers to whether impingement or manual cleaning, manual wiping action, spraying as well, cascading flow on the side of the vessels, or agitated immersion at the bottom of the tank. So what are the cleaning agents you're going to use? Cleaning agent can have different type, whether alkaline or acidic, depending on the type of soil that you have. Some even need the booster as well for those most challenging soils that some customers may have. And the concentration, temperature, all this water quality, needless to say, important in the cleaning parameters. And weather factors and the number of rinsing as well. So you wouldn't want to use too many rinses at the end of the cleaning to remove the soil and also the cleaning detergent. So selection of the appropriate cleaning agent to the respective soil is very important in the design of the optimized cleaning parameters. So these are the parameters affecting the cleaning performance. Of course, the surface of the process equipment, the product contact surface where the soil is. So the surface property, the material of construction, and also the equipment design. Make sure that the equipment is designed to be easily cleanable. And also the surface must be smooth as well. Of course, a smooth surface will facilitate the removal of the product residue from the surface. If the surface is corroded, for example, rouging or corrosion occurs, that will basically impact the cleanability of the surface. So the validated cleaning procedures may not be effective to remove the residues from those surfaces. And cleaning parameters, as I mentioned before, the cleaning parameters, the time, the action, chemistry, what detergent to use, concentration of the detergent, and also the temperature as well. So these are critical parameters: water quality, dirty hole time, and also the toxicology data for the cleaning agent if you're using the cleaning agent. Soil characterization as well, soil residue, you need to understand the kind of soil that you are having and going to remove. So all this will need to understand and design in the design of the effective cleaning parameters. So this is a case study number one. This is a multi-product facilities for the finished products. In this case, the product evaluated contains a polymer and an insoluble oil. So a polymer is basically a long chain of monomers. So this can be considered as a macromolecule, a big molecule actually. So it's not easy to clean in most of the cases. Of course, the insoluble oil, as you all know, not easy to clean as well, because it's not soluble in water. So in this case, basically, lab scale studies using 304 stainless steel coupons. So if you're using the sodium lauryl sulfate, so this is one kind of cleaning detergent. If you use 2% concentration, and this 15 minutes, 60 degrees, there'll be trace residues observed on the coupon after the cleaning. If you're using the formulated alkaline cleaner, formulated means there's surfactants, chelation agents added into the formulation. So 3%, 60 minutes, 60 degrees, still have some trace residues. So in this case, because this soil is not easy to clean, so the formulated alkaline cleaner will need to be used in conjunction with the detergent additive containing hydrogen peroxide. So basically, this can be considered as a booster for the alkaline cleaner. So in this case, it's 1% alkaline cleaner plus 1% considered as a booster. There for 15 minutes, 60 degrees, then it's visually clean. So from here, you can see that, I mean, for the most difficult to clean soil, it's very important to use the appropriate cleaners and boosters, if necessary, to remove the residues effectively and in a short time. So, so this is basically the benefits of having a lab-scale studies to work out the cleaning parameters for the most challenging soils that any facilities have. And then the case study number 2, this is a cell culture medium. So in this case, of course, a cell culture medium contains a lot of media as well as the cell debris. Sodium hydroxide, if you use sodium hydroxide alone, not effective in the cleaning process. Acid detergent, not effective as well. There's a light residue. If you use the alkaline formulated detergent, 1% is effective for the removal of this soil. And then if you try to reduce it to 0.5%, it's also effective. So the benefits of this lab-scale study is it enables you to basically also optimize the cleaning parameters. Although 1% can be cleaned, but you can basically check whether a reduced concentration can be effective as well. So this is the benefits of the lab-scale studies on the cleaning of the soil. So this poll question number 1. So Christina, can you pull up the poll question? Okay, so this is the question for you all. So the question is, do you perform cleaning cycle development or optimization? So there are 3 choices. Yes, by lab scale studies, followed by cleaning trial runs using the actual equipment. Second option is yes, by cleaning trials in equipment directly without the lab scale studies. Third one is no. No can be you just use whatever cleaning parameters that was passed down from previous cleaning process or other cleaning products. Yeah, so you do not perform any trial run, just use whatever cleaning parameters that you have for the cleaning. So just give you a few minutes to provide an answer to this whole question. Okay, let's look at the result. So from the result, 25% perform a lab scale coupon studies followed by cleaning trial runs, 49% by doing the cleaning trial runs in equipment. Okay, and then no is 25%. Yeah. So most of you basically perform trial runs in equipment. Yeah, so that can be done as well, but yeah, you will need more time because you do it in actual equipment. You need more time. And when you want to adjust the parameters, yeah, you will need more time as well. You have more equipment downtime. So that can be done as well. Of course, the lab scale studies will be the best approach. It allows you to optimize the process quickly and then scale up in your actual equipment. Yeah, so so this is the result of the poll question. Okay, let's continue. Okay, next we look at the critical performance parameters. So in the cleaning process, of course, cleaning temperature, the time, turbulence flow, or the action in the piping, especially the pipe as well. You need the turbulence flow for the cleaning action, cleaning agent concentration, and also the process time, including the dirty hole time. The CQAs for the critical quality attributes. So visual inspection, very important. After cleaning, you need to ensure that the equipment are visually clean for those parts which can be visually inspected. And also you need to identify any API residues specification by calculating the limits using the macro. If you're using detergent, of course, you need to include the detergent in the calculation for the macro as well. And also any micro contaminant specification will need to be included. So for the sampling methods, of course, during the cleaning validation, sampling is very important. You typically have swab and rinse samples. So swab samples, of course, the physical removal is good because it's very technique dependent. You swab on the specific locations. Of course, hard to reach locations. Sometimes you cannot reach every part of the equipment. Invasive as well. You need to dismantle some of the piping if you want to swab certain areas in your process equipment. So for rinse, it's basically it's not technique dependent because it's just cover the whole areas of your process equipment, including irregular surfaces. It's non-invasive, can be used on the online monitoring. So these are some of the pros and cons using swab and rinse. So if possible, both should be used. And recovery studies for swab, you need to perform some recovery studies. You have the swab methods and the rinse method as well. You can perform the recovery for the rinse using, by rinsing the coupon surface, and then you check the recovery of the residues through the rinse. So these are the two typical methods that can be utilized for the recovery studies for the rinse. Of course, the determination of the swab locations is very important. So any swab locations on your process equipment need to be justified and documented. The sampling method as well, the plan materials to be used for the sampling location and the sampling. Of course, in your cleaning validation, you need to specify exactly the location of your swab samples. So detection methods can be specific, for example, HBLC methods, or non-specific can be TOC methods. Microbiological requirement as well. So these are the typical swab locations for the vessels. So worst case locations need to be specified with the rationales. Typical one is basically the agitators, the outlet valve, some of the sampling ports, the side glass as well. So this can be justified, by the low velocity of cleaning solutions or poor contact cleaning solution, or high potential for soil retention, especially the ledges in those sampling ports or the instrument ports. So this is basically the locations and the sampling methods for the API cleaning residues. Some assessment. Sampling locations can be the sidewall, bottom valve, dome, instrument port, and the agitator. So the criteria you can look at is the potential for the large containment area or any hotspots, historical hard to clean areas, affinity to the material construction. So sometimes some soil is stick more to certain material constructions, and the role in the process likely to lead to difficult residues. So here is because it's the same vessel, so everything will be the same. Of course, you need to look at the cleanability of the location as well, the coverage and the accessibility. So for example, the sampling port, the instrument port will be sometimes the coverage or not easy to clean. So just do a ranking, and of course this highest ranking will need to be included in the swab locations. So these are just examples of the things that you can do to determine the swab locations. Product grouping, especially for the multi-product facilities, you need to select the representative products for the cleaning validation. So typically the conditions for product grouping needs to be similar product type in the same equipment train, and must have the same cleaning process using cleaning detergent, cleaning methods, and the process parameters. So must be the same cleaning process. Then you can group the products together and select the representative products for the cleaning validation. Of course, some of the criteria can be considered is using the high potency, high toxicity, or allergenic products, and also some conditions that is unacceptable to consumers, like for example, the fragrance particulate as well, hardest to clean or remove. So as mentioned before, the justification for worst case residues can be based on the cleanability. So how easy to clean using the cleaning solution from the surface, and the toxicity, solubility of the product as well in the cleaning solution. So these are examples on the product grouping. So for example, here I have 3 products. So drug active, number 1, have calculated residual limits of 25. And then product number 2, 15. Product number 3 is 30. So option 1 for cleaning validation, you can validate the product number 1 with the lowest calculated residual limits. So you just choose this combination for the cleaning validation. Of course, option number 2 is to validate 2 times 2 products. So validate product number 1 with 25 microgram of the residue limits, and then also you validate the product number 2 with the 15 microgram of residual limits. So there are 2 options in this case basically. So typically you will need to choose the most difficult to clean and also the lowest residual limits of the product group. So these is the examples on the strategy approach. So for the equipment group, so if you group equipment, if you have different cleaning procedures. So for each cleaning procedures for the equipment group, you have various products. So from here, you will need to determine the worst case macro, and then also determine the worst case product to clean. So that is the criteria for choosing the 3 runs from this equipment group that is using the cleaning SOPs number 1. So this is just a warning letter on the cleaning validation, recent warning letters. So basically the FDA expectation is that you need to choose the worst case for the product cleaning validations. So this can be based on the higher toxicities, higher drug potencies, drug of lower solubility in their cleaning solvents or cleaning detergent, and the drugs with characteristics that make them difficult to clean. So for example, the enteric coatings and things like that. Soap locations for areas that are most difficult to clean, and also the maximum hold time before cleaning. So 30 hold time longer will be more difficult to clean. So this is just a flow chart on the cleaning validation. So when you run a cleaning validation, this is run 1. So first thing is to soil the equipment, and then you have the dirty hold time, for example, 3 days. Then you run the cleaning cycles, you collect the rinse samples, and perform the visual inspection and the soap sampling. So if any of this fails, then you will need to have a deviation to investigate. Investigate the reason for the failures. So if you pass, then you just perform the second run and the third run until you meet the required number of successful cleaning validation runs. So this is just an overview of the typical cleaning validation flow chart. So of course, the process capability and limits. So when you have the cleaning process or cleaning validation, when you have enough data, you want to make sure that your cleaning process is capable to meet the acceptance criteria consistently. So this is a good way to ensure that your cleaning process is capable. So you can look at the chart basically. So this is the limits, and if your cleaning process is very low, very low compared to the limits, for example, 30 to 40%, or even 20%. So that will be a robust cleaning procedures to have, because that will ensure that you can consistently meet the acceptance criteria. Of course, you can set some alert and action limits based on standard deviation to alert you when there's an adverse trend happening during your cleaning process. So that will give you an early warning to perform an investigation into the root cause. Okay, last slide here. Basically, the cleaning validation, if you have the out specification, will need to be investigated. Of course, must have a thorough investigation, whether is it due to equipment or the cleaning process not robust, or there's a human error. So you need to have the root cause determined before you repeat your cleaning validation. So completion of all cleaning validation runs implies the cleaning procedures are effective in reducing the products to acceptable levels. So going forward, after post cleaning validation, you need to make sure that you ensure that the validated cleaning procedures continue to operate in the validated state by having the controls in place, such as the change control approvals and also the monitoring annual review of the deviations. So with that, I end my presentation here, and I will hand over to my colleague Paul to continue on the health-based exposure limits. So Paul, I will stop share right now.
Hey, Richard, thank you for that detailed part on elements of cleaning validation and a great summary for that. I'm going to move forward now with a review of regulatory timelines for health-based exposure limits. And then I'm going to go into some supporting documentation around health-based exposure limits. And lastly, applying some of these health-based exposure limits in regards to cleaning. And so the first thing is just to kind of indicate that the process of removing the contaminants from the process equipment to maintain the condition of the equipment, such as the equipment can be safely used to make subsequent products. And Richard talked about that, multi-product facilities requiring cleaning validation, understanding what is the risks to the next product being produced. And when we're talking about a contaminant, it's the presence of a minor ingredient in another chemical or mixture, often at trace levels. So very small amounts on the surface. And cleaning is a subjective term. So we need to look at defining this and setting appropriate acceptance criteria for this. So what is an acceptable level of residue limits? Um, and so what we need to do is to determine how much of the product we just cleaned, so generally product A, would be administered in each patient taking the next product, so product B. But in order to make this number useful, we need to determine how much that will represent in the next batch, and then translate that to a number, to a value in terms of how much that will be present on the surface to measure. And that could either be a direct surface sample or through a rinse sample analysis. And we also need to understand a safe level. And this is where in the past it used to be applying safety factors. Now we're using health-based exposure limits to determine what is a safe level that could be carried over. So when we think about what is an acceptable level of residue, how clean is clean comes up. And every once in a while, I hear there's zero residue on the surface from product A, whether it's active or excipients. And that really is not a realistic amount. The limits must be rational and acceptable. And it must be able to detect your lowest limits. And a couple of things that you want to kind of watch out for is basically setting arbitrary limits. So linking them right to compendial water specs, uh, linking them to the analytical assay that's used for your method, maybe selecting it arbitrarily or unrelated to target residue, such as using 10 parts per million. And then also not having documentation in regards to that rationale or understanding of how they were selected. So those are some things to kind of watch out for. Now you can go through and set health-based exposure limits and then default to a compendial water spec or default to the analytical method that's used, um, or default to, you know, an expected value based on your process capabilities. But you do not want to immediately go to that approach without having an understanding of the risk of that previous product to the next product that's being produced. And how we're going to determine the basis for quantitative limits. Now, the traditional approach, and this is based on the FDA guidance in 1993 and the Foreman and Mullen article, looked at a carryover of 10 ppms or 1/1000th of a therapeutic dose and visually clean. That's referred to now as kind of the traditional approach. You could also use the toxicity of the residue based on a toxicity endpoint like a NOEL, no observed adverse effect level, or a low L, or even in some cases um, uh, uh, an LD50, and I'll kind of go over what's the differences between these different values. And then we have a health-based exposure limit, which reviews the pharmacological and toxicological effects and determines a PDE value or an ADE value. And I'll go into those acronyms as we move through the presentation. So the first thing we're going to do is do a poll question to kind of see where the audience is with this. And Christina or Linda, if you could go ahead and display the poll question, that would be great. And the question is, how do you, oops, I might have to move this to the screen so people can see it. So how do you obtain the PDE value for the products manufactured? The first one is engage a qualified toxicologist. Second one is based on an occupational exposure limit, which is kind of a manufacturing of the ingredient. And then work out the values based on any available toxicity data on your own. And then the fourth item is default to 10 ppms and visually cleaned. So I'm going to go ahead and select one. And we'll wait about a minute to see what the answers are, and then kind of proceed with um, the regulatory timeline. Okay, so this is quite good. We have 43% of the participants engage a toxicologist in determining a health-based exposure limit, or in this case, a permitted daily exposure value. 14% is based on an occupational exposure limit. Occupational exposure limits are generally determined as a safe limit for the workers manufacturing the product. And then 20% work out the values based on any available toxicity values on their own. And we'll talk through some concerns with that approach. And then the fourth item is default to 10 ppms and visually clean. We have 24% of that indicating that. And we want to be a little concerned in terms of what is the potential carryover. This may not be an issue for um, uh, low toxicity products that we're making, it may be a bigger concern if we're making things like oncology drugs. So I'm going to go ahead and close that and then move into the rest of the presentation. So when we talk about the limit timeline, and this slide's a little bit busy, but I think it's a nice way to visualize that. We can start off with the 1993 guidance that Richard talked about, and then the Foreman and Mullen article that talks about the traditional method of setting limits. And then we go into the PDA Technical Report 29, again, a document that Richard talked about. And then we then move into 2010, which is where the ISP baseline guide came out talking about risk-based manufacture of pharmaceutical products. And this was primary the initiation of use of health-based exposure limits and determining assessment based on toxicity assessments by a toxicologist, and that is to deal with some of the highly potent drugs that were starting to be manufactured, especially around oncology drugs. And then that was kind of moved forward with the EMEA guide on setting health-based exposure limits in 2014. And they introduced the term permitted daily exposure, or PDE value, in 2014. And then we have the adoption of that in 2015. And then we go into the international, or what used to be the American Standard Test Methods guide for derivation of health-based exposure limits in 2020. We have the PICS aide-memoire in terms of inspection of health-based limits. We have the World Health Organization working document on points to consider regarding health-based exposure limits, and the Brazilian Anvisa technical note in terms of health-based exposure limits. So you can note in 2020, we had 4 documents published to interpret the use of health-based exposure limits that was originally introduced by an acceptable daily exposure from the ISP guide to a permitted daily exposure through the EMEA, and then in trying to add some more details through the ASTM, PICS, Anvisa, and World Health Organization. So just to kind of recap, ISPE, EMA, PICS, ASTM, World Health Organization, and Visa all advocate the use of health-based exposure limits, and specifically in the terminology of accepted daily exposure, or an ADE, originated with the ISPE risk map, and the use of the term PDE, or permitted daily exposure, was introduced through the European guide in 2014. The general term is health-based exposure limits, and it refers to a limit based on a toxicological evaluation, and it's focused on how carryover might cause harm to the patient that takes the product. And it represents a dose. It's generally in, mgs per kg per day type of value. And again, it's unlikely to cause an adverse effect if an individual is exposed at this dose every day for a lifetime. I do have, and I'm not going to get into the fine details between the acceptable daily exposure and the permitted daily exposure. That is provided to you with this. But I do want to point out, a couple of fine points with this. The ADE was based on any route of administration. So if you see a term that's used as an ADE, then it's taken into account whether it's a parenteral product or a topical product. Um, if you have a PDE value, the true definition of that doesn't, it allows for multiple PDE values based on a route of administration. So that's really the main point to think about when you're talking about an ADE and PDE. The latest World Health Organization, as well as in Visa, refer to these as relatively equivalent terms and used interchangeable. But I just wanted to point out those two items with that, and note that the body weight is referred to as a 50 kilograms for human medicinal products and 1 kilogram for veterinary medicinal products. The weight can vary based on the anticipated recipient of the product. Now, when you're working off a kind of a point of departure, it's based on a toxicity evaluation, and it's generally based on a NOEL value, um, which is a no observed adverse effect level. And it's really is at the very bottom of a dose response curve because you're not seeing any observed effect, adverse effect. As you're moving up this curve, you get to, a no observed effect level, a low L, which is a low observed effect level, and a low L, which is a low observed adverse effect level. And then you get to an LD50, which is a dose uh, that results in death of 50% of the population. When you go into the European um, uh, Aide Memoire, it indicates do not use LD50 values to set health-based exposure limits. So what I recommend, if you're seeing an LD50 value, try to look at the original study and see if the study actually had the dose that killed 50% of the population. If it did not, then um, look at interpreting that as a low L or no L value based on the results of that study. For the past 100 years, we've been using LD50 values to look at toxicity of pharmacological products. So again, if you're seeing LD50 values, take a look at that's the original study to see what that, if that's truly is an LD50 value or another determination on that. So what we have is we have a point of departure, that's that no L value, and it can be a low L value as well. There's an adjustment factor for that. We have the weight adjustment that's typically 50 kilograms. It could be lower if it's a veterinary product or maybe for a newborn. And then we have a series of factors. Um, and in the ADE approach, they use uncertainty factors. In the PDE approach, they use F factors, and they account for things like extrapolation between species, variability between individuals, maybe account for repeat dose toxicity, um, in regards to, in case of severe toxicity, or adjustments based on whether or not a no L or a low L was used. And then sometimes you can have some pharmacokinetic factors as well placed onto that, or modifying factors. But you have some visibility in terms of uh, that adjustment factor that's used. In the past, that was referred to as a safety factor. Now you have a series of factors that represents the rationale for it. So what type of supporting documentation helps in terms of that health-based exposure limit? And this is the case if you're provided a value, maybe from an organization or maybe from one of your company sites, in the sense of when an auditor comes in, they may ask for, how was that number determined? And so it's good to have a clear understanding to how that was determined. So with this in mind, let's take a quick look at that World Health Organization um, uh, guidance around health-based exposure limits, and specifically around cleaning agents, which is myself and Richard's specialty. Um, so the cleaning agent should be selected with care. This should be appropriate um, for its intended use. And the selection of the relevant cleaning agents should be scientifically justified. There should be some proof of effectiveness of the cleaning agent, and there should be some points to consider would be what concentrations are used, as well as the composition and removal of that residue to an acceptable level. And lastly, the cleaning agents are used in the cleaning process. These should be included in the cleaning process development studies and cleaning validation. So this was kind of in line with, what Richard was talking about with lab evaluations. We're seeing definitely a pickup in terms of laboratory work to support the selection of cleaning agents, as well as the selection of cleaning parameters that are then taken to the field trials or to the cleaning validation um, process. Now, with that in mind, a lot of times cleaning agents are formulated products, so it's a good idea to understand what the components are and also how the components rinse from a surface, because that's going to drive your analytical method to show that they've been removed from the surface to the safe levels. So this is just a study that was done actually over 20 years ago, but it's a formulated cleaning agent. Multiple methods are used to look at the different components, and you're understanding the rinse rate of those components as a function of the analytical testing. So all the components rinse at pretty similar rates, so that a number of different methods could be used to demonstrate that in this case, this cleaning agent has been rinsed from the surface. And so that when we go into establishing a health-based exposure level, or an ADE or PDE value, what we want to do, and this is specific to a cleaning agent, it could also be from an active, we want to collect all the toxicity information available. And that's from safety data sheets or toxicity studies. We want to understand any raw materials that are used, as well as the product formulation. And then we want to get that to a toxicologist to identify a point of departure, and then assigned factors associated with that. So here we have, in this case, we have a NOEL value of greater than 120 mg per kg per day. And that's based on an intravenous toxicity study. And we have the adult male body weight of 50 kilograms. And then we have our daily dose, which is that NOEL value times the body weight of 6,000. We then, the toxicologist then assigns an uncertainty factor based on the intraspecies, interspecies, subchronic, low L to no L, as well as the database completeness. And again, the UF values are out of the ISPE risk map for the ADE, and we get a cumulative uncertainty factor. In this case, it's 900. And note that it does have a range specified, a default value, and then the selected value from the toxicologist in assigning that. And again, this is determined by a toxicologist. So kind of pulling that all together, we have the point of departure, or the NOEL value. We have the body weight. We have the cumulative uncertainty factors. And then we have what's called a modifying or pharmacological adjustments. And then we have our final ADE or PDE value, and that's in mgs per day. And again, this is the amount that can be exposed given any route of administration over the lifetime of exposure. And here is just written in a slightly different way that looks at the uncertainty factors written out. So with obviously all those documents around 2020, we're we myself, Richard, others have received a lot of questions in regards to supporting information for that. So here's some information um, that was to look at a report that was issued in 2015, which is a year after the original um, document from EMEA, and it indicated that reference report on the information that was provided was prepared by a certified toxicologist. It was also reviewed by internal subject matter experts from tech services and regulatory affairs. It is available under confidentiality through a quality system audit, and that there's some information in regards to table of contents and the copy of the approved cover letter. So again, you're thinking about what supportive documentation around that value that you've been provided. Um, and so some of the internal reports would be things like the point of departure, as well as any toxicity information that was reviewed as part of the package. So in this case, there's dermal toxicity, aquatic toxicity, acute uh, intravenous toxicity, as well as oral toxicity, and so forth. You're indicating what was actually reviewed by the toxicologist. And the report is signed again by the toxicologists as well as subject matter experts internally. And then you get into, you know, the descriptions around the uncertainty factors. And again, this is done by the toxicologist as well as um, and reviewed by the subject matter experts. So why was the interspecies uncertainty factor established? What was the interspecies uncertainty factor established as? Is there any sensitivity, any mutagenicity, carcinogenicity type of things to keep in mind? Is there any subchronic to chronic extrapolations? And was the low L and no L views and so forth? But you have now clarity in terms of why that value was assigned for that specific uncertainty factor. And then you have generally a sketch or complete CV of the toxicologist on record so that that can be reviewed as well, to make certain that they have credentials to establish that health-based exposure value for um, that that active or that excipient, or in this case, that cleaning agent. So in terms of what I was talking about from supporting documentation is completed by a certified toxicologist. You identify hazards and characterize them, identify any critical effects. You determine a point of departure, typically a no observed adverse effect level or a no L value, and then you establish uncertainty factors or F factors, as well as pharmacological and modifying factors. And then you have the calculation, and then that report is reviewed and approved by internal subject matter experts. Um, and so this would be supporting documentation around the health-based exposure limits, and that's in line with INVISA, World Health, EMEA, and so forth. Um, so let's spend a few minutes and talk about applying this. Um, so this is just a different way to look at what Richard presented. So we have the different stages of the cleaning validation on your left, and we have a list of potential hazards associated with the product. Obviously, active ingredient, API, is up there on the top. Cleaning agent I highlighted on the bottom. Those are two things we normally think about, but there's an awful lot of things in between that could be a worry. We always want to review the engineering and design, which Richard talked about, and coverage testing. And then we always want to think about the parameters and method selection. And then we want to think about what do we want to use during the validation to support the routine monitoring. That could be surface sampling and recovery. It could be visual inspection. It could be rinse sampling and recovery. We want to choose the method that makes sense to demonstrate that the surface meets a pre-established acceptance criteria, so it doesn't adversely impact the next product being produced. And you can use different techniques here, providing that they correlate to a surface cleanliness. So when we talk about residue limits, there is a number of different ways that we can define the limit, and we'll go through some of that. It could be a daily amount allowed. It could be concentration in next product. It could be maximum allowable carryover. It could be amount per surface area or per swab or per rinse volume. There is a number of different ways to define limits. So it's good to make certain that you have that clear, as well as understanding of the units that are used. And just a quick example here, we have product A. We want to be thinking about the minimum daily dose of product A. In this case, it's a tablet manufacturing. So we're going through the tablet, in this case 50 meg tablet, and the number of tablets that can be taken per day. It gives us a minimum daily dose of 100 megs per day. Now, when we go to product B, we have 1,000 tablets, 1 tablet per dose, 2 to 4 doses per day. We can then go ahead and calculate that to be about 4 grams per day. So that as we look through the calculation, we have 100 mgs per day of product A. We have a PDE value that's been assigned. And then we have the product B, the maximum daily dose of product B at 4 grams per day. And we get a concentration in the next product of 0.025 mgs per gram, which comes out to 25 grams per gram, or 25 ppms. Now, at this point, you understand what is that value, and now you can then go ahead and default to a value. So this is where, in this case, they're looking to default to 10 ppms, which is kind of based on the previous traditional method. And then you can determine the maximum allowable carryover by understanding the batch size. So the batch size is 20 kilograms, is 200 kilograms, and we determined that we're going to use the 10 micrograms per gram, and that gives us 2 million micrograms. Then we can go ahead and take the 2 million micrograms and divide it, in this case, by the shared surface area of 450,000 centimeters squared, and we get now a limit per surface area. So 4.4 micrograms per centimeter squared. We can then adjust that based on just that for the swabbing, based on the size of the swab area that we're going to take, whether it's 25 centimeters squared that's used in this example, or 100 centimeters squared. We can make some adjustments to that. So in this case, we have the 4.4 micrograms times the 25 gives us 110 micrograms of product A for a swab sample of 25 centimeters squared. And then we also should be thinking about the extraction volume. So in this case, we have 20 grams of solvent. We want to take that into consideration. And now we have 5.5 micrograms in the swab extract. And then we can also adjust the swab extract to adjust that limit by increasing or decreasing that volume. So in this case, we went from 20 grams to 5 grams, and we increased it from 5.5 to 22. And this is important if you have methods that are not sensitive enough to what you set. You can make some adjustments to the swab area as well as um, the diluent volume. And this is where you can hear increasing the swab area to make some adjustments um, to do that. So this is going from 25 centimeters squared to 100 centimeters squared. And you can see if you took 100 centimeters squared swabbed and utilized 5 grams of extract, you would get a limit now of 88 micrograms per gram. So again, this is all linked to that analytical method and being able to show that it meets your acceptance criteria. And this is just to kind of correlate with what Richard had in terms of some grouping. In this case, we have 4 products, 4 different actives, 4 different limits that we utilized. If they're all cleaned the same way, then you can use product D, active P to the lowest limit of 10 micrograms. If you have, if product A is the hardest to clean, then you want to use that cleaning procedure and then set it to the lowest limit of 10 micrograms. So as there's no, those are just 2 options for grouping. There's a lot of different ways to group, get people involved within the organization and establish how you want to group. And then I would rethink that based on the numbers of samples and what types of work resources are needed to validate that based on that grouping approach. A couple more things to kind of add, and I need to be mindful of the time, is the MEA guideline talked about um, if the active ingredient is degraded as a function of the cleaning process, then you may not need to look at a toxicological evaluation. And that's clearly in the EU guidance. So just a couple of examples of this. We have a therapeutic macromolecule. Um, it's known to degrade. We have different downstream manufacturing process. We have different cleaning agents that are used, different cleaning times, different observations that are observed, and the time that it took to clean that material. Now, we also, so essentially a 1% alkaline detergent, 5 minutes was able to get a visually clean surface. When we go and look at evaluation the degradation, that 1% at 5 minutes at 45 degrees, we see that we have an intact molecule. And then when we go to 1% at 60 degrees, which is in lane 3, we also have a relatively intact molecule. When we go to 3% at 60 degrees for 20 minutes, we have essentially over 90% degraded. So here we're looking at a cleaning process to remove the residue from the surface at 1%. But if we want to degrade the molecule as a function of the cleaning process, we're looking at a higher concentration. And then in this case, we have our controls at different concentrations for comparison. And this is a monoclonal antibody around 140,000 kilodaltons. When we go to another approach, a small molecule approach, we may have a different strategy here. So here we have small molecule degradation. A 1% alkaline detergent was essentially, and this is using um, NMR. We have our control, 3 nice peaks. We have an alkaline detergent, was very effective at cleaning, 1%, 5 minutes, but we have a degradation of the active ingredient. So therefore, it changes potentially our toxicity profile. It also changes whether or not our analytical method is going to be acceptable. However, if we clean with a neutral product at 2% for 30 minutes, we have an intact molecule, and now we have our analytical method and our toxicity evaluation can be effective. So utilize that degradation information on determining the performance of the analytical method that's used, as well as the toxicity information that you have in setting your limits. And at this point, I'm going to kind of wrap up. Um, is we talked about some elements of cleaning. We talked about history of health-based exposure limits. We talked about supporting documentation around health-based exposure limits. And lastly, we provided a couple of examples, including looking at degradation of a macromolecule, as well as some concerns with degradation of a small molecule. And the degradation of the macromolecule, typically using total organic carbon to show that it's been removed, so that it really doesn't impact that method to show that it's been removed. And again, adjusting the concentration can make a big difference in terms of degrading that macromolecule. So at this point, I think Richard and I will open it up to any questions that the group may have.
Oh, there's one question from the chat box from Cornimex. So the question is, should we choose the lowest value of NOEL if there are more toxicity data, for example, the dose, repeated dose, reproducibility, carcinogenicity, or various test animals length of study? So that is the question.
Yeah, I mean that's a challenging question, Richard. And I mean, I think what we want to be thinking about the choice, it's really the point of departure. So you have all this toxicity information. It's a matter of choosing a tox study and a NOEL value that's generally representative of the route of administration and the that you would be uh, that the active or the hazard would be found. And again, that's determined by a certified toxicologist. And then you base your uncertainty factors based on that point of departure that you selected. So as you're talking about multiple toxicity studies, multiple NOEL values, you really want to look at choosing one as the point of departure. So as the example that I had, there was actually multiple intravenous toxicity studies done on that cleaning agent. The toxicologist selected one of them as the point of departure and then moved into determining what the uncertainty factors from that.
Okay, thank you. Yeah, there's another questions. So how do we define or choose the certified toxicologist? So is there any criteria or qualification for certified toxicologist?
Yeah, I mean that is a tricky question, um, Richard, and I think with that, it's really take a look at the CV on the toxicologist. And with that, I would even evaluate the subject matter experts that are reviewing and signing off on the report. And that is a qualitative assessment that's done. But you want to be looking at obviously education as well as experience with those types of calculations and hazards. And there's several of them out there. I would go back to the ISPE risk map and then move forward from there. And there's most recently that ASTM guide is also very detailed in regards to setting limits. But you know, anytime that you look at qualifications, that is kind of subjective. But that's why it's important to have the CV or biosketch in reference so that it can be reviewed and make certain that they're qualified. Now, some companies utilize multiple toxicologists and then base a value, and that could be based on the lowest value It could be based on a consensus of all of them. Again, you're justifying your approach to that health-based exposure value.
Okay, thank you. I think there's no more questions. Oh, there's one more question, I think. How do we figure out PDE when we have the NOAEL of I in monkey study only?
Wow, that is extremely specific. Well, I mean, one is you're using that as a point of departure. So, okay, you have that study done in a monkey, which is a very good animal model, especially if you're using for an ophthalmic product that you're manufacturing. Then you want to then kind of move forward with your uncertainty factors associated with that. So that would be kind of interspecies, intraspecies, as well as completeness of your toxicity database. And obviously working off one study, you have a limited toxicity database, so you want to probably adjust that value accordingly. So again, is check with the toxicologist and then move forward with the studies that you have available.
Okay, next question is, what are the professional services and equipment that STERIS can provide in the areas of cleaning validation and toxicology? So for example, consulting equipment. So for the cleaning validation wise, of course, STERIS, we supply the cleaning detergent for the customers in the areas of process equipment cleaning. So in the event that, the customers have some questions or require technical support in the use of the detergent for the cleaning validation, we can always provide technical support. In terms of the troubleshooting or the optimization of the cleaning parameters, and also some guidance on how to determine the cleaning limits for the detergent use. So in those areas, we can provide the technical support. This is where we will help in the cleaning validation aspect. So Paul, you want to mention a little bit about the toxicology support?
Yeah, so obviously providing PDE or ADE values on cleaning agents, as well as support around setting limits for active ingredients like we've shared in this presentation. Those are some of the things that we would be looking at doing. There was the next question. Richard talks about the PIXS, mentioned about potency besides cleaning and solubility. This is one of those things, and Richard covered this earlier in terms of grouping. So generally you look at potency, toxicity, cleanability, solubility in terms of grouping products to determine a worst case. What we want to do is, in terms of health-based exposure limits, is understand how that relates to the worst case product when we're looking at setting the limits. So again, the potency, cleanability, and toxicity, cleaning and solubility was referenced specifically around grouping, and that is identifying what's a worst case product or active.
Okay, so next question is specific method to measure the carryover always mandatory in every case? Or is there any justified case that we can only use non-specific methods using TOC limit, for example? So basically, it depends on the type of soil that you are considering. The specific method is always preferred. Because they are very specific. And of course, in those cases whereby, for example, if you have a protein-based products, so normally after the cleaning process, the protein can be degraded. So in those cases, you can't use a specific methods to detect the carryover. So those cases, you can use the non-specific methods such as the TOC methods for the detection of the residues. So it really depends on the type of products that you are looking at.
Yep. And the only thing to add to that is if you're using a non-specific method, then you have to look, it's going to not differentiate between the active ingredient in Richard's example, or a non-active ingredient in the formulation that contains, in this case, organic carbon, or the cleaning agent. So keep that in mind when you're using the non-specific method, but it can definitely be used to determine an acceptable limit.
So the last question is, what is the acceptance criteria for visual requirement?
Yeah, so the visual requirement can be a qualitative test, so you want to define the procedure for doing that inspection. And if you are looking at setting that to be more quantitative or a limit test, then it's preference to do some spiking studies of the acceptance limit on the surface, and then qualify operators based on distance and lighting and angle and use of eyewear or goggles or face shields, and then determine what is the limit that can be visibly detected given those criterias. And there's some documentation around that, and the article that was referenced included visual inspection as well as surface and rinse sampling, and it's a good resource for that. Yeah, the last question, and we'll kind of wrap up, is, what's the acceptance for the limit of a microbial swab and rinse? There's a good reference for that Richard provided, the PDA technical reports. They talk about 1 to 2 CFUs per centimeter squared as kind of a good reference point for that. I always take into account the type of product that I'm manufacturing, as well as the route of administration, and whether or not I'm picking up objectionable organisms as well when determining a microbial limit, whether it's a swab or rinse. So we did not cover that in today's presentation, but you want to take into account what's the product, that is, whether or not it's preserved or not, what's the likelihood of growth of the organism, as well as, again, the route of administration, the patient population, and whether or not there's any type of existing condition like immunocompromised, in setting appropriate microbial limits for surfaces or rinse samples. Great questions. And I think at this point, Richard and I will bring it back to Linda and Christina to wrap up the webinar.
Thank you very much, Paul, and to Richard, and to everyone who's stayed on for a few minutes extra this morning. Just to thank you both for your time and expertise, as always. It's a pleasure to have you, and we look forward to another session in the new year. For everyone that's joined and those that joined late, we will be sending the link to the PowerPoint as well as to the recording for you after this session. If you have any questions, please do feel free to ask the speakers as well as the team from STERIS. I know that Ike and Jenwei are on this recording, but they will be able to follow up with you directly. Also, you are very welcome to view the recording on our ISPE Singapore YouTube page, alongside all the other recordings of the STERIS workshops that we've been holding over the last few years. So we look forward to seeing everyone again soon. In the meantime, wish everyone a great day. Paul, a good evening. And thanks everyone for joining. Take care, everyone.
Okay, thank you. Bye-bye.
Thank you.
Thank you.